So hello everyone, thank you for joining us today on our webinar, Data Transformation and Cluster Analysis in FCS Express 6. As I mentioned, this webinar is being recorded and will be posted to our website and our YouTube channel. My name is Juan Vasquez and I am the Customer Support Manager for Novo Software. I am joined on the line by Allison Irvine, who is our Scientific Algorithm Software Developer, and by Andrea Velle, who is one of our Technical Application Specialists located in Italy. So FCS Express 6 now allows you to use data transformation features to quickly define, access, and use data transformations such as tSNE, K-means cluster analysis, principal component analysis, or PCA, parameter math, as well as giving you the ability to integrate R scripts for your data analysis. Today's webinar, we're going to try to be covering all of these different transformations that are available in FCS Express 6. So, some of the topics that we're going to talk about is why would we use FCS Express for data transformation? Well, hopefully from today's webinar, you'll be able to see that the overall interface of FCS Express is easy to use and intuitive. Hopefully it comes naturally to you. But what you'll also see that a lot of these transformations are going to be significantly faster than their counterparts in R or in other third-party softwares. So some of the transformations that are available now in FCS Express 6 are going to be the T-Distributed Stochastic Neighbor Embedding, or tSNE, K-means cluster analysis, R integration, and the ability to do parameter math that we'll talk about later, and PCA, or principal component analysis, has been available in FCS Express since the last version, the last major version, FCS Express 5. Okay, so without further ado, what I'm going to do is I'm going to transfer the mic to Allison Irvine, who's going to be giving you a mathematical background about tSNE. Okay, Allison, it's all yours.
Okay. Hello, everyone. I should probably go to the beginning of this presentation. Okay. Here, I'm going to give a short introduction to the background of tSNE, which is short for T-Distributed Stochastic Neighbor Embedding. So I'm going to start, though, by talking about dimensionality reduction. tSNE is a dimensionality reduction method. So the basic problem that we're trying to solve here is that we've got this really high dimensional data, and we can't see all the dimensions at once, but we need to visualize the data in some way that's meaningful. So quick background, there's different ways that people do dimensionality reduction all the time. Feature selection is one major way of doing it, and a lot of people do it manually using their biological knowledge, their background knowledge, to say, oh, well, I know that the information I'm looking for is actually all able to be visualized in these two parameters, so I'm only going to show these two. That's actually a form of manual feature selection. Of course, there are statistical ways of doing it where you can try to determine which dimensions have the most information in them. That's what people have probably done the most, but we're going to deal with feature extraction, which is where you take your data and you actually try to derive new parameters from your existing parameters. And we've got two major fields, linear dimensionality reduction and nonlinear dimensionality reduction. So here's a little example to illustrate what I'm talking about. So I've taken this data that is all the way here on the left. So you can see there's actually 4 stripes of data that are somewhat separated by space. And this is two-dimensional data right here. And what I've taken is I've basically pretended like it's on a piece of paper and rolled it into a C shape. So now I've kind of artificially created a third dimension by just rolling this up into a C. And so you'll see I've tested a linear dimensionality reduction method, PCA, and a nonlinear dimensionality reduction method. So PCA basically takes that C and views it from some angle. It like takes this three-dimensional data and smashes it down onto a flat surface. And so you end up with this, which doesn't really give you much information. It doesn't really help you like see these 4 populations that are in here. But if you use a nonlinear dimensionality reduction method, it will actually do a closer examination of the underlying structure, which is what we also call the manifold. That's this C shape right here. And it'll try to learn the structure of the data along this curved shape. And so it actually ends up basically unrolling this C and almost recovering that. So tSNE falls under the nonlinear dimensionality reduction category. But as you can see here, it's actually a little bit different from most traditional nonlinear dimensionality reduction methods, in that it highlights and exaggerates the local structures. So the reason it's useful for our purposes, flow cytometry data, is because we're actually not really interested in necessarily learning the overall underlying structure. We're actually interested in learning in finding clusters of data. So tSNE does a good job of that. It actually takes these parts that are, these sections of data that are clustered together, it actually exaggerates this by pushing them closer together. And it takes these gaps in between the data and exaggerates those and pushes those farther away from each other. So what you see is these like more tightly clustered groups. So it's actually pushed these together into little more circular shapes. And that's why it's useful for us. So here's just a screenshot from FCS Express. You can see I've got some high parameter flow cytometry data. And there's some populations that you can see if you go through and try to gate it in different dimensions. But not all of them are separable in all of them. So tSNE has taken that. We've done a dimensionality reduction and extracted 2 parameters from all of these parameters. And it's kind of taken these and pushed them all together, you know, all the cells that are close together, and it's also tried to like create this segmentation between the distinct groups by exaggerating the spaces in between them. So here's a really short overview of how the algorithm works. So the algorithm first starts with the input data, the high dimensional input data. It calculates a distribution over that data by taking the distances between all pairs of points. And then you randomly initialize your new tSNE data, your new 2-parameter data. And then you do this iterative thing where you calculate the pairwise distances of your tSNE data, you create a distribution, and you try to match it to the distribution of the input data. So you're just going back and forth between steps 3 and 4, and you're just searching for 2-dimensional data whose distribution of distances between data points most closely matches the distribution learned on the input data. Now, some people, if you've heard of tSNE before, you'll hear people sometimes say that the results of tSNE can be different each time you run it. And that's because of these 2 key points right here. One is that the tSNE data is initialized randomly. So every time you run tSNE, it could potentially be different data that you're starting out with, and then you're shifting the points around until you end up with something that's close to it. The other point that I want to make is this minimization. So your distributions aren't going to match exactly because you've got 2-dimensional data and some, 8-dimensional data or something, but they are going to get as close as possible. But there's many ways in which they could be as close as possible. So here's just a little bit more illustration. So tSNE will start out with this like random little tiny cluster of data to get a distribution. It'll start shifting the points around, spreading the points around over and over and over again until you met some maximum number of iterations. And each time it's spreading these points around, you're getting closer and closer to this input data distribution. Now we run 500 iterations, through some manual experimentation. Usually 350 might actually be good, but 500 is pretty sufficient to be safe. Between 500 and 1,000 iterations, you don't usually see much fundamental change, as in like cells going to different populations or new populations appearing. Usually you'll end up with some group of populations and they'll be kind of shifting around in that 2D space. But those structures will still be there, generally speaking. So we leave it at that. Here's some options. So these are the options that we've given to you guys. We've got your basic options. We've got a template file, so that's where you choose your data file that's got the parameters that you're trying to apply tSNE to. So this just kind of helps you figure out what parameters you have available, and from there you'll see these checkboxes of your available parameters in your original data, so you can select which ones you want to reduce down to 2 dimensions. You can apply a gate to the input data, so you'd be only performing tSNE on whatever data is inside your gate. We've got a choice here now between which tSNE method you want to use. You can use exact tSNE or a Barnes-Hut approximation. So exact tSNE is what I described to you just now in the algorithm, where you're calculating the distance between every single pair of points in your data space, so that you really know where everything is. Now, Barnes-Hut approximation is a method that approximates the distributions over your data. So we apply some tricks that basically, instead of having to calculate the distance between every single pair of points, you can use a subset. It's much faster. It's definitely worth it if you're running tSNE on more than, say, 1,000 data points or something. Now there's a sample size. Oh, sorry. The amount of approximation ranges between 0 and 1, and this only applies to the Barnes-Hut approximation. So 0 is the least amount of approximation, at which point it's actually slower than exact tSNE, so you may as well just use exact because they're pretty much the same. But 1, all the way up to 1 is the maximum amount of approximation that you want to have. Usually 0.5 is decent, is going to give you decent results. Now there's a sample size, and so this is important because this algorithm is very time-consuming and memory-consuming. So I'll show you a little chart of running time, so you can help make that decision on how big you want your sample size to be. And what we do is we randomly sample some of the data, we run tSNE on that, and then optionally you can estimate tSNE for the unsampled events. So what we do is we kind of take all your data that wasn't sampled, and we kind of find its nearest neighbor in the original data space, and we estimate the unsampled data's tSNE coordinates based on the sampled data's tSNE coordinates. And then there is a little checkbox here to say automatically calculate tSNE. So that's to turn on and off live updating. So if you have this box checked, and you change, for example, the amount of approximation, tSNE will automatically recalculate on your plots if it's been applied. However, if you have that box unchecked and you start changing parameters or whatever, nothing will happen until you actually click on the Calculate tSNE button. So you're not waiting in case you're running a large sample size and it's going to take forever. So this is a little bit more elaboration on exact versus Barnes-Hut. So like I was saying, exact tSNE calculates the distances between every pair of points to create the distribution. So that can get really expensive, right? If you have, so if, you've got 1,000 data points, the amount of data required, that's 1,000 squared distances. So that can grow pretty fast. And can get really slow and can eat up a lot of memory. So we use Barnes-Hut as an option. And this is just some details on how that works. The input data distribution, we do the distance between each data point, its nearest neighbors only. And on the tSNE data, as we're iterating, it does like a probability, something like a probability binning, similar to that, and then you're basically, instead of calculating the distance between every point and every other point, you take each data point and you calculate the distance from that to the bins. And the approximation parameter is actually affecting that particular step. So the approximation parameter determines something like the bin resolution. So if it's 0, it's like you're gonna, every cell is in its own bin. If you have it at 0, you're gonna calculate the distance between the point and every single bin, which is equivalent to every single cell. If you have more questions about this later, you can ask. I've got some extra slides too on some more details. So here's the last slide on running time. And you can see this is the time in seconds. And this, the red line here is the R implementation of tSNE from the package tSNE. The blue line is our FCS Express version. We've done a few like little like optimizations with like threading and data management and stuff like that, memory management that helps us out a little bit. And here's the Barnes hut. So you can see when you start getting up to what is this, 10,000 here, 10,000 samples, you're already at 4 seconds in FCS Express. And then here, you're at 150 seconds for 5. So for 50,000 samples, right? So if you're running it on a reasonably fast computer with 4 cores, I didn't ask Andrea actually what kind of computer he ran this on, but it's reasonable to say you can probably run up to like 100,000, 500,000 without any like major problems. Once you start getting past there, it starts taking a really long time. So it's worth using that subsampling and then just mapping the unsampled points later on. A lot of times you're going to get really similar results as long as like all your populations are can be sufficiently represented by the number of samples. It's going to be okay. Few extra notes I want to say before I pass it back is that try to make sure that you've applied any variant stabilization or normalization before you run tSNE on it. And try to make sure the scaling is like pretty similar for each parameter. Okay, I'm done.
All right, thank you, Alice. That was excellent.
Yeah.
Okay, so what I'm going to do is I'm going to show you how we implement it here in FCS Express. So I'm not necessarily going to focus on the creating plots or different gates or anything like that. I'm going to assume that everyone here that's joining this and watching this webinar knows that. But if it turns out you would like more information about creating plots or creating gates or getting statistics from different populations in your analysis, whether it's just a regular plot or from a tSNE plot or whatever, please feel free to go to our website, denovosoftware.com, and we have a whole library of different videos and webinars that have been recorded and have been placed on there that you can view at your own leisure. So right now what I have here is just an example type of analysis where we have a data set, and what I'm going to do is I'm going to try to apply a tSNE transformation to this to show you how it's all integrated within the software. So the first thing I'm going to do is I'm just going to go to our Tools tab. So for those of you that are familiar with previous versions of FCS Express, the Tools tab is new. From within the Tools tab, we'll have a Transformations window that is now available. And within the Transformations window is where we're going to add the transformation of interest, such as tSNE. So once we add that, it's going to be selectable here in the window. And then we'll see that we'll have a number of different parameters that can be edited for this, as Allison had described. So the way this works is we would select a template file, and if we already have a data file loaded, FCS Express will use that as a template, and basically what it's using as a template is just the parameters are saved with the file. We would select the parameters that we want to include in the transformation, whatever gates are available, and whether we want to use the exact tSNE or the Barnes hut approximation, okay? So the default approximation that we use is going to be 0.5, but again, if you want to drop this down to 0 or increase this up to 1, that's up to you. You have that, that possibility here. And then we can also adjust the sample size. Right now, FCS Express is defaulted to include all the unsampled events within the tSNE calculation. If it turns out you did not want that, you can simply deselect it. But at this point, if you want to calculate the tSNE, you can just simply calculate tSNE, and the approximation should happen here in the background. Okay, so what I'm doing here is I have a combination of color dot plots, some density plots, some contour plots that just have different coloring patterns to them. But what I want to show you is the fact that on these particular tSNE plots, we can create gates, and we can gate different clusters of interest. And using a lot of the tools that are available in FCS Express, we could emphasize those events. So as I create a gate, I have the ability to emphasize that, and that means that I can increase the dot size for those events. And we can see these in other colored dot plots, so we can actually focus on where a particular cluster is in a two-dimensional space where we have an X parameter and a Y parameter that's specific for the fluorochromes that you've collected or any of the parameters that you collected with that data file. There's a number of different gates that we've created here, and they all act the same. So we can move this around. You can see that as I move this around, even within the transformation, all my data is updating automatically as I move these. So we can actually see this as I move my gate, even a pixel, I can see all the data update immediately. Okay? And the same theory applies to either the contour plots or any other plot type that we have available to us in FCS Express. And we can use any number of tools that we want to help calculate data that we're deriving from these particular transformations. Okay? You can see here that it is significantly fast. So this is backing up what Allison was pointing out, how we are significantly faster than the same tSNE counterpart that's run through an R script or through any other tSNE implementation that's done through any of our competitors. And using a lot of our tools that are already built into FCS Express, you can simply just use this to help analyze any data that's being transformed through a tSNE application. Okay. So on that note, what we'll do is I'm going to shift it back to Allison, and who's going to start talking about Kmeans, which is another transformation that is available within our transformations window. Okay, Allison, go ahead.
Okay, hi. So we're going to move on to clustering now. So at a very high level, clustering is just a process of grouping data so that all the cells or all the data points in one group are the most similar to each other. They're more similar to each other than to the data in any of the other groups. So it goes under the category of unsupervised learning, meaning that you don't need any pre-classified data, you don't need any prior knowledge about your data to perform clustering. Clustering is somewhat of an exploratory data analysis, like, you know, you're looking at your data, you want to see, okay, well, let's see how my data naturally groups together, and then we'll find out if that's meaningful. You know, using either a computer model, you know, doing some applying some extra algorithms afterwards, or you can even use your own background knowledge to kind of determine what the clusters mean. So we're going to talk about Kmeans today. It's going to be a short introduction. It's the simplest, pretty much the simplest clustering algorithm that there is. Most people learn about this one at the very beginning, and it's a great concept, though. So it's a centroid-based clustering. Centroid meaning, you know, like the mean value of all of our data points in a cluster. And so the entire principle runs off of data points. You have a set of centroids, one centroid for each cluster, and your data points are just assigned to the cluster with the closest centroid. So we use Euclidean distance to define what is closest. And so this is kind of how it works. K-means, because we're using Euclidean distance in this way, it's actually assuming that the clusters are all spherically shaped. Now I'm showing this in 2 dimensions, but you can imagine, you know, these could be spheres or hyperspheres as you go up in dimensionality. But see, I've got these centroids here, here, and here. And these circles I've drawn around them are kind of like, they're just like an equal radius around the centroid. So everything, any data point that would lie along this circle would have a distance of 1 from this centroid, a distance of 2 from the centroid on this circle, and 3. And so what you end up actually is kind of like a decision boundary where these circles intersect. And so everything, you know, these lines that I've drawn where the circles intersect, everything on this side, any data point on this side is going to end up being assigned to this cluster. And any data point on this side is going to get assigned to this cluster and so forth. So the algorithm, in a nutshell, works like this. You start with a random selection of centroids. Then you assign each data point to the centroid which is closest. And then you recalculate the centroids based on which data points were assigned to which cluster. So you can see this green dot moved over here because that's actually the average of the points that were assigned to this cluster. And then you just go back and forth between steps 2 and 3 until the data points stop switching clusters. So kind of made a little thing. This is how this works. So like this is just running k-means through several iterations, read it from left to right and then down each row. So we start here. You can see these random centroids. We assign all the data points to clusters. And then this centroid moves up here, and this centroid moves over here. So now you got your new centroids in the next iteration. And then we reassign all the data points. And the centroids shift around a little bit more, and you can see over time, they all slowly converge so that the centroids slowly move so that they're all kind of at the center of these clusters that naturally occur in the data. And there you go. And that's your result. So k-means is found in a lot of places because it is fast, it's easy to implement, it's easy to understand, most importantly, and it's really easy to also describe the clusters that you end up with. All you need is a centroid value. You don't need really any other information about your clusters other than the centroid. Like if you want to use it as a model, for example, and assign new data points, all you need to keep is the centroids. However, there are some disadvantages because you know, it is so nice and simple and easy that there has to be something something wrong with it. So well, one is the common problem actually. You need to specify the number of clusters. And then it doesn't perform well with non-spherical clusters. If your clusters have weird shapes, you've got an L around a circle or something like that, it's not going to perform well. If you can't separate your clusters with straight lines, it's not going to perform well. And it can also converge to a non-optimal result. Similar problem to tSNE actually. K-means is also generally randomly initialized. Although people do, and it's also like minimizing something. It's minimizing this, it's minimizing the distance of all the points to the centroids. So you've got this random element and the minimization element. So therefore you end up with this kind of, the results can be different depending on how you initialize the data. Now there's like a lot of existing heuristics that people use to kind of deterministically say, well, okay, I want the centroids to be initialized in this way, but it really, the results are going to depend on whichever method you use, whether it's random or some guess that you made based on your intuitive knowledge of the data. But it is extremely useful still because of its ease and because of the simplicity. It's used very often as a pre-processing step or an intermediate step for other algorithms such as SPADE or Kmeans. It's used to initialize other clustering algorithms such as like Gaussian mixture models, EM or something like that. And sometimes, you know, these other, a lot of other algorithms will find ways to like overcome some of these obstacles by incorporating other steps to try to guess the right number of clusters, or to get around that limitation of the spherical clusters by over clustering the data, like learning too many clusters, and then starts like combining the clusters together in some way, which is actually what both Spade and Kmeans pretty much do in different ways. So, last couple slides. So in FCS Express, we've got a couple ways of visualizing your clustering results. One is with histograms. So this on the right is my original data. And I've applied Kmeans clustering to it. And I'm visualizing the clustering results in the histogram. And what I've done is I've backgated. So I've drawn markers on the histogram and then color-coded the dot plots according to those markers. So that way you can see the dots colored according to the clusters in your dot plot. So we've got, this is histograms where it's like each cluster, and it's histograms of the distance of all the data points within that cluster to the centroid. So you can see some small differences in the spread of the data within the cluster. Here's another way of seeing it, though. We've got little Gaussian histograms. Each one is a little Gaussian, but they're scaled to the sum of squared error, kind of like the variance of each individual cluster. So the clusters that are more spread out are going to be wider here. And like M2 is kind of a narrow cluster, so you'll see its histogram is a little bit narrower. And then this one down here, it's basically the spread of these histograms is not scaled, but it is scaled by the number of events. So like, the height of the histogram of each peak is going to be proportional to the number of events within the cluster. Oh, sorry. And there's another way we've done it. It's very similar to the last slide, except that we've kind of generated some random Y data so that you can view the same data from the histogram. You can view it on a dot plot as well. You know, if you prefer drawing circle gates or you know, polygonal gates instead of a marker. So this is the same information down here. It's this is actually where they're not scaled at all, and they're just going to show up as equal-sized round clusters. This is the same thing, though. The SSE scale, that's the same as this histogram right here, except I've just added some random data up here so you can see it nicely on a 2D plot. And that is the end of my presentation. I'd like to switch it back over.
All right, thank you, Austin. So we will have Andrea Valli present on how to use Kmeans within FCS Express. So Andrea, go right ahead.
Yeah, thank you. I think now you can see my screen.
Yes.
So we just see Alison just present us what Kmeans. So let's see how we can use Kmeans clustering in FCS Express. So this is my example, my sample. What I'm going to do is to perform Kmeans on this gate that I call my gate, based on a bunch of different parameters that I will select among the available parameters in this file. So first, let's open the transformation dialog. As one previously showed us, if you go in the Tools section of the ribbon bar and click in the Transformation button, the transformation dialog will appear. And you will be able to select the transformation that you want to create, in this case, Kmeans. As you can see, the dialog is pretty similar to the tSNE one. Actually, it's similar also for other transformation like PCA or R integration, and this is because we try to be as consistent as we can. This way, you will familiarize more quickly with this transformation dialog. So, again, on the left, you have a list of parameters, and on the right, you have a bunch of different options. So the first step is to retrieve the list of parameters, and actually you can do this by selecting a template file. In this case, the template file is already selected since I have this file already loaded in my layout, but you can simply click on this ellipse button and select a new file as a template. Now you can select the parameter that you want to use for your K-means clustering. You can select a gate and the number of cluster. Let's say we want 3 clusters. Then you have a couple of other options, for example, the maximum number of iteration. As Alison told us, K-means is an iterative process, so this is the maximum number of cycles after which K-means will be stopped. So if you don't have a specific reason to change this parameter, you can just keep it as it is. And then you have this other option, the minimum % cells per cluster, which is actually the minimum size of each cluster. For example, if you set this to be 5, this means that each cluster will contain at least 5% of the events. Finally, you can decide the output that you want to use. The K-means transformation in FCS Express is able to generate different K-means parameter. So by default, the cluster assignment is selected, but if you want, you can select also other K-means parameter. I'm not going into detail in this case. I will just refer to the K-means manual. If you go on our website and actually on our manual, you will see that we have an entire chapter dedicated to transformation, and here in this K-means chapter you will have all the definition of the available K-means parameter. But for this demonstration, I will just assign some of them, and I will show you how you can present these results using heatmap, histogram, and two-dimension plot. So now that we select the K-means and the K-means parameter that we want to generate, you can simply drag and drop your K-means transformation on top of your object. For example, on top of your heatmap, on top of your histogram, and on top of your two-dimension plot. And you see that the title of this object actually reflects this change. So you see that this K-means clustered suffix has been added to the title of each object. And this tells you that the transformation has been applied. Very briefly, the cluster assignment parameter is the only one which is compatible with heatmap. You see here, each cluster is actually represented as a well in this plate. And for those of you who are not familiar with heatmap, let's consider that the heatmap is a very useful tool, since you can display your results in a very comprehensive way, and also you will be able to color each of these well, and thus each of these cluster based on a given statistic. For example, in this case, you see here the y-axis say CD4 median. It means that the color of each cluster gives us information about the MFI calculated as median of the CD4 parameter. So you see, for example, that cluster 2 has the highest CD4 MFI among these 3 clusters. And you can change the parameter just by click and select another one. And in this case, you can see that it's cluster 3 as the highest CD8 MFI among these clusters. Also, if you go in the Format section of the ribbon bar and click on Overlay, you will be able to change not only the parameter used to color your cluster, but also the statistic. For example, you can decide and select one of these available statistics. Also, just consider that in version 6, in FCS Express 6, you can also decide to change the radius of each well and thus of each cluster based on another statistic. So just keep this in mind. All the features that are present on heatmap can be used also in this case with the K-means transformation. Finally, you can create well gate, and thus to create gate that contains all the events that belong to a given cluster. Just right-click on your heatmap and select Create Gate Well. Then you have to left-click on the well that you want to be a gate, and let's select a name and, for example, a color. And then click Close when you want to apply the change. You see this gate, this well gate now it contains all the cells within this well and thus all the cells within this cluster. And I already create the other two well cluster. So now you can use this gate for your downstream analysis. So you can apply it to other plot, you can create new plot gated on this gate, you can backgate, whatever you want. So this is how you can display the cluster assignment using heatmap, but as I told you before, you can also use histogram or two-dimension plot. You have just to select one of these other 5 K-means parameter, and once you apply the transformation on your object, this parameter will be accessible as standard flow parameter. You see here, this is one of the parameters that also Alison showed us a couple of minutes ago. So the unscaled, each of these peaks is actually a cluster, and we decided to allow you to use this representation because in this way you will be able to draw a marker and to retrieve statistic referring to each cluster using a simple histogram. And of course I'm not going to do it, but just consider that you can convert and link a marker to gate, and so you will be able to use that gate for your downstream analysis. Last but not least, you can also display K-means parameter on two-dimension plot. So first, you can just select one of the K-means parameter and you can combine it with a standard flow parameter, for example CD8, and you see that cluster 3, which is this one, actually contains CD8 bright cells, and this is this goes together with the information that the heatmap gave us a few minutes ago. Or you can also combine a K-means parameter with another K-means parameter, and this is the Gaussian Y data that Alison mentioned a couple of minutes ago. So this is just to allow you to plot clustered events in a user-friendly way on two-dimensional plot. But anyway, the idea is that you can create gate on your two-dimensional plot and then use this gate to create other plot or to apply this gate to other plot and histogram. So the idea is that you can easily manage the clustering results using all the objects that are within FCS Express. So K-means is a really good way to familiarize with clustering, but of course if you want to perform let's say a more complex clustering, or if you want to apply a transformation which is not listed here, for example a transformation which is not tSNE, what you can do is to exploit a new feature in V6, which is actually the R integration. So let me simply switch on this other layout. R integration is a way that allows you to integrate and run R script directly within FCS Express, and actually to be able to use the results of this script again within FCS Express. So a very brief introduction on how to enable this R integration. So the first step is for Windows user to go in the web page of the R project and download the R statistical software. This is not required for Mac user. The second step, and this is required for both Windows and Mac user, is to enable what we call the the Novo software external application bridge, which is actually the way in which FCS Express can communicate with R. So both Windows and Mac user have to go here in the user option under the startup category, and you see this, the last option is start the Novo software external application bridge on login. So by default this is not flagged, so this is not active. So please, if you want to use the R integration, just flag this option as I already did it, and then click OK. So I cannot click OK since it's already selected, but if you select it for the first time, then you will be able to click OK. After this, you will have to completely restart FCS Express. And once you restart it, you will see that in your icon tray, you will see a new icon, which is this one, the Novo Software External Application Bridge icon. And this will tell you that the bridge is properly running. So this is a very brief introduction on how the R integration is working. I mean, it's not mandatory for you to know this, but I think it's interesting. The idea is that your data will be exported as a matrix from FCS Express to R. R will execute your script, and the results of your script will be re-imported in FCS Express and will be available, for example, as a new parameter or as a cluster assignment. So up to now, we have two prototypes of script that can be integrated in FCS Express. So the first one is R add parameters, and this is suitable to add new parameters. For example, if you want to create new transformation and you want to add the result of this transformation as new parameters. So more or less the same that tSNE does. So to create new parameters, the input of this script will be the matrix with your data, while the output will be an object that will contain the new data, the number of new parameter, and the name of the new parameter, nothing more. The second prototype is the cluster transformation. Again, the input is the matrix containing your data. The idea is that this script will generate a cluster assignment as a result. So the output of this script will be the cluster assignment, so one per each event, the number of cluster, and will also contain the name that you want to give to your cluster. So what I'm going to show you, it's a very easy, basic example of the R add parameter prototype. And in my script, what my script does is just to apply a log transformation to the input parameter. So for example, if you select the forward scatter as an input parameter, the output parameter, so the transformed one, will be just the log transformation of this. So this is just a very basic example, but you can apply any kind of super advanced function to your data. So again, let's open the transformation dialog and let's select the R add parameter transformation. Again, you see that the dialog is pretty similar to the other one. You have to select a template file. This is already selected since this file has already loaded in my layout, and you can select a bunch of different parameters on which you want to perform your transformation. The second step is to select your script. So this is my script. Then you can select a gate if you want to perform the transformation only on a subset of cells. In this case, I want to apply my transformation on the entire data set. And last, you have to decide the orientation of the matrix, so the orientation that you want to use to export your data from FCS Express to R. And this depends on how you build your script. For example, I build my script using a matrix oriented with events as columns. So I have to select this option. And then as for the other transformation, you can just drag and drop your transformation on top of one of your plot, and you see that the R add parameter added is reported in the title. This means that the transformation is applied. So as you can see here, you have three new parameters. So the log forward scatter, the log side scatter, and the log CD3 fetch. So in this plot, I applied the transformation. In this other plot, I didn't apply the transformation, and you see that the parameters are just the original one. So it's very easy, and if you click on a new parameter, you will see that this new parameter will immediately appear in the list of available parameters. So, and just to show you that the results is correct, so I'm displaying here the two of the new parameter, log forward scatter and log side scatter, and on the left side I will just change the scale of this plot. Instead of linear, I will use the logarithmic scale. You see that the picture is exactly the same, but if you look carefully, you see that the non-transformed parameter are actually plotted here, and you see that the original value, 10 to the 5th, while on the plot on the right, we have the two transformed parameter, and you have indeed the log results of this transformation. So it's very easy, and of course, you can create, you can plot the new results as a two-dimensional plot, as a histogram. You can create marker, gate, and so on and so on and so on. So this is really a way in which you can easily integrate your script into FCS Express, and that's to combine the statistical power of R and the power of graphing the results of FCS Express. Just a brief summary, we have two prototypes of script that can be integrated in FCS Express, the add parameter and the clustering transformation. Finally, we will soon have an R package for the R integration in FCS Express that will further simplify the writing of script that can be made compatible with FCS Express. So I think now it's the turn of Juan that will show you a very brief overview of other two transformations, which are actually the PCA and the parameter math.
Thank you, Andrea. That was excellent. Yeah, so we're going to wrap up the webinar with our final two transformations, parameter math and principal component analysis, or PCA. So just quickly, so the parameter math is a procedure where we can create simple data transformations that use mathematical operations to create a new parameter for our data. And then we can use that parameter and display it on the X or Y plot axis within a plot that we're using within our analysis. So again, if I just open up my transformations window, if I just click on my plus sign, you'll see that one of the options available is parameter math. So if I simply select the parameter math, what I'm going to do is I'm simply just going to create the formula down below that I'm going to use to create this particular new parameter. So I'm just going to click on new, and when I click on new, I'm going to simply give this new parameter a name. In this case, what I'm going to do is I'm going to create a ratio for a calcium flux experiments. So we're going to give it a name. And then we have two ways of how we can define this formula. It could be a simple formula where we take one parameter, choose the mathematical operation, and then we choose the second parameter. Or we can also make a formula where if I right-click in here, we can use one of any number of different tokens or functions that are available to us in FCS Express. So for example, if I actually wanted to take a parameter and convert it from lin to log or from log to lin, I can choose a similar function and use that in there and just update the variables according to my data. In this particular case, what I'm going to do is I'm just going to use simple. And what I'm going to do is I'm going to take FL1 parameter, divide it by our FL3 parameter, and then click OK. So then you'll see that here in my formula sequence, it's going to show me the equation I've created plus the two different parameters that are being used in that operation. So if I want to apply this to a plot, I could just simply take this parameter math, drag and drop it. You'll see that in my title, it'll tell me that my parameter math is applied, and the new title, the new parameter will be available on one of the axes. This one hasn't been applied yet. You can see that there's no parameter, but again, I could either drag and drop, or I could simply format this. And from the Overlays tab, I can choose the transformation that I want to use. And then again, you'll see that that new parameter is available here as well. Okay. And then finally, just going to talk about PCA quickly. And as Allison had mentioned earlier, PCA is a linear dimensionality reduction that projects our data into a flat surface. And again, this PCA transformation is similar to all the other transformations where we'd simply just add it to the list, select it from the list, and then we'd format the parameters that are being transformed down below, where we have a data file that's being loaded. We'll actually look in that and analyze the different parameters that are in there. We would select the parameters that are to be included as part of this transformation. We would select the gate that's going to be included for this transformation, the cells within that gate. It's going to be included in that transformation. And then we're going to choose the number of parameters that are going to be selected for this transformation. So this number 3 just means that we are going to have 3 different parameters that are going to be added to these files. And once we do the transformation, we'll have those parameters selected here for us automatically. So if I actually click this and make this 4, then you'll see that I now have a 4th parameter, and this all updates live. Okay. And again, this all updates in real time. So as I move these around, you'll see that all my data is going to update. All the new clustering is going to be updated as well. Any colors that I've had will automatically update as well. Everything happens live in real time. Okay. So in summary, I think this is a good time to wrap it up. I know we're running against the clock here, but in summary, the data transformations in FCS Express are optimized for speed, and we've presented the functionality in a easy to use and intuitive interface where we just simply open a window, we add the transformation of interest to that list, and then we're just going to format where we select the type of parameters that we want to be included in the transformation, the particular cells that may be located within a certain gate, and the type of transformation that we want to use. And we can simply just apply it to a plot through a drag and drop process, and then just click on the X or Y axis in that plot to choose those new transform parameters. We have 5 transformation types that are currently available, as we discussed, and we are hoping to implement more in the near future. So if you do want more information about these transformations, we have many different resources and examples on our website. So I encourage you to visit our website, denovosoftware.com, to see that information, those resources. If you have any questions, you can always email us directly at support@denovosoftware.com. And you can also visit our website, either denovosoftware.com or fcsexpress.com, to download a free 30-day trial where you can try these transformations out with your own data and see how you like it. And if you have any questions with using these new transformations, please feel free to email us and we'll be happy to help you. Okay, so I'm going to stay on the line for about another minute and we'll take any questions that come across. If there are no questions, thank you for joining us today. I want to thank Allison and Andrea for joining us today and for doing an excellent job presenting these new transformations. Again, if there are any questions, I'll stay on line. Otherwise, thank you for joining us and have a great day. Okay. As I mentioned, if you have any questions, please feel free to email us at support@denovosoftware.com. I'm going to end the webinar. Thank you for taking the time to join us today, and I look forward to hearing from you in the future. Thank you and have a great day.